Right now, in your chest, a muscle is quietly performing a continuous rhythmic miracle. But how did it get there? Welcome to This Explainer. I am absolutely thrilled you're tuning in today because we are going to unpack one of the most mind-blowing biological journeys in existence. We're looking at the mammalian heart, not just as an organ, but as a masterpiece of biological engineering. We're going to trace its formation from a tiny, seemingly random cluster of cells to the vital pump that literally sustains your life. It is, for all intents and purposes, a process of anatomical origami, folding and twisting itself into absolute perfection. But before we get into the nuts and bolts of the mechanics, I really want you to ponder this. How does a flat, relatively simple embryo manage to build a highly complex, four-chambered beating pump from absolute scratch? It's a profound biological puzzle. See, as an embryo grows, it very quickly reaches a size where simple cell-to-cell diffusion just isn't enough to deliver nutrients anymore. It needs a dedicated delivery system, and honestly, it needs it fast. Okay, let's dive into this. Here is our roadmap for today. We're starting with humble beginnings, watching two tubes become one, exploring the great heart twist, building four chambers, checking out the fetal bypass, and wrapping up with the miracle of birth. All right, let's start right at the beginning with section 1. The heart's humble beginnings. So as the embryo is rapidly developing, its growing mass of cells creates this really urgent biological problem. It desperately needs a functional vascular system to move nutrients and oxygen around. To solve this, the embryo turns to a specific embryonic connective tissue. Let's bust some jargon right here. Both our blood vessels and our blood cells arise from a tissue called the mesenchyme. Within this mesoderm layer, specifically in the wall of the yolk sac, the very first formative tissues appear. These are specialized cells known as angioblasts. You can think of them as the foundational building blocks, basically the raw plumbing material for our entire cardiovascular system. And what is utterly fascinating about this, no way around it, is that the plumbing is actually built before the pump even exists. Groups of these angioblasts clump together to form solid masses called the blood islands of Pandor. Now, these solid clusters don't stay solid for long. They soon begin to hollow out. As they do, the cells on the outside flatten to become the endothelium, that's the walls of the blood vessels, while the cells in the center differentiate into the very first blood cells. And from the wall of the yolk sac, this incredible network of microscopic pipes extends right into the body of the embryo. With the preliminary piping all laid out, we move right into stage 2. Two tubes become one. Our focus now shifts to a region located in the splanchnic mesoderm, known as the cardiogenic plate. This is where the actual primordium, the earliest structural hint of the heart, really starts to take shape. But here's the catch. Initially, the heart isn't a single structure at all. It actually starts out as two completely separate endothelial tubes. As development marches on, these two tubes slowly move towards each other and fuse, first at the front and then at the back, to form a single central tube. Once they're fused together, the walls of this single tube differentiate into three distinct layers. The internal lining becomes the endocardium, the surrounding mesodermal layer transforms into the thick muscular myocardium, and the outermost layer becomes the epicardium. And this brilliantly illustrates exactly what we're talking about. Looking at these transverse sections, notice the foregut at the top and the pericardial column down below. Right there in the middle, you can physically see how those separate structures merge together. That simple, single tube, suspended by the dorsal mesocardium, represents the earliest unified heart. By simple constrictions alone, this primitive tube is already dividing itself into three regions: paired atria to receive veins, a single ventricle, and a single bulbus leading out to the ventral aorta. So, how does this simple tube become a heart? Let's check out our third stage, the great heart twist. Guys, this is without a doubt the most dynamic physical transformation of the entire embryonic process. The heart is about to undergo some really severe contortions. The absolutely crucial thing to understand here is why it twists in the first place. The heart tube starts growing at a much faster rate than the pericardial cavity that surrounds it. Because it's trapped in this confined, tiny space, the rapidly lengthening tube simply has nowhere to go. It has no choice but to buckle, fold, and bend upon itself, contorting into this tightly spiraled S shape. It's like trying to stuff a 10-foot garden hose into a shoebox. Tracing this folding process right here, you can see that as growth continues, the bulboventricular loop drops backward and downward. This incredibly precise spatial folding takes that single straight tube and stacks it. It brings the primitive atria and ventricles into their proper anatomical alignment, setting the physical stage for the next massive phase of internal construction. And that brings us perfectly to Section 4, Building 4 Chambers. If that previous step was like bending the foundation of a house, this step is like a frantic contractor rushing to put up the interior walls, all while the water is literally already running through the pipes. The heart manages to pinch and divide its single inner space into 4 distinct chambers, using a series of developing internal walls, which we call septa. The 2 upper atria are separated by the development of the septum primum and the septum secundum, which eventually fuse together. Meanwhile, down below, the single ventricular space is subdivided into a right and left ventricle by the interventricular septum. At the very top, 2 endocardial septa fuse to carefully split the common bulbus into 2 completely distinct exit pipes, the aorta and the pulmonary artery. But here is where mammalian biology throws us a massive, brilliant curveball. Welcome to Section 5, the fetal bypass. Now, what's really interesting about this slide is the unique problem the fetus faces. Because the embryo isn't breathing air yet and its lungs are essentially just filled with fluid, it uses the ultimate fetal hack, mapped out right here. Highly oxygenated blood enters the embryo through the large umbilical vein. It travels straight up to the liver, mostly routing directly through a vessel called the ductus venosus, to hit the posterior vena cava. It mixes with a little bit of venous blood there before arriving directly at the heart. To avoid pumping all this precious oxygen-rich blood into the fluid-filled, non-working lungs, the fetal heart utilizes 2 ingenious bypass valves. First, we have the foramen ovale. This is a literal hole between the right and left atrium. Most of the highly oxygenated blood from the lower body shoots straight through this hole into the left side of the heart, completely bypassing the right ventricle, and gets pumped straight up to the developing brain and head. Second, we have the ductus arteriosus. For the blood that does accidentally make it down to the right ventricle and into the pulmonary artery, well, this specialized vessel shunts it straight over into the descending aorta, once again bypassing the lungs and sending it down to the rest of the body and back out to the placenta. Which leads us to the grand finale, Stage 6, the miracle of birth. This perfectly balanced, incredibly delicate fetal system remains in place for months, but it is all building up to the climax of our narrative where everything changes in literally a matter of seconds. Contrast the situation before and after birth. Before birth, the placenta provides the oxygen and the lungs are entirely bypassed. But the very moment the baby is born and takes that first dramatic gasp of air, the lungs suddenly inflate. This creates a massive, instantaneous shift in pressure within the chest cavity. The placental circulation violently ceases, immediately throwing those large umbilical vessels into complete disuse. The newly functional lungs suddenly demand blood, and the entire circulatory roadmap is rewritten in an instant. Because of this sudden pressure shift, the temporary fetal plumbing is forced into a structural lockdown. The flap of the foramen ovalis snaps shut, permanently sealing the right and left atria apart, leaving behind only a small depression known as the fossa ovalis. Furthermore, that bypass vessel we talked about, the ductus arteriosus, it quickly atrophies and is transformed into a solid fibrous cord called the ligamentum arteriosum. Even the umbilical vessels and ductus venosus dry up into ligaments. Just like that, the adult circulatory system is fully online. As we wrap up this explainer, I want you to just take a second to feel the pulse in your chest, or maybe your wrist. When you consider the delicate, incredibly precise perfection required to fold this embryonic origami from single angioblasts to simple merging tubes to a twisted S-loop, perfectly partitioned with septa, and utilizing ingenious fetal bypasses that permanently seal the exact moment you took your first breath,